Hello, everyone. So I'm Kritika, and I'm a PhD student in University of Tasmania, and I work under supervision of Professor Tim Broadtrip, Dr. Jules Freeman, Dr. Ibrahim Barbia, and Dr. Rebecca Johns. And my study is actually part of a bigger project. It's a research which is supported by just a second, please. Yeah, so it's a research which is supported by New Zealand Ministry of Business, Innovation and Employment, and it comes under a bigger project which is entitled as Seeing the Forest for the Trees, Transforming Tree Phenotyping for Future Forest. So today I'm going to talk about the first study of my PhD, which is about how the stomatas in Pinus radiata, they respond to VPD. And before heading to my main study, I would talk a bit about the background. Okay. Yep. So if you look at all of these 5 studies, you would find something in common. And that's the major focus of the studies. So all these studies, they aim to improve the transpiration use efficiency. And there is one more common thing about these studies. So if you look at these studies carefully, you would realize that all of these studies, they actually focus on the crop plants. Now, the question is why the trees they are actually missing from these studies. So this is picture of from one of my field experiments where we are actually fitting some optical dendrometers on pine trees. And here we cannot use weighing balances. So weighing balances are not an option for for actually measuring the transpiration use efficiency in terms of big trees, which we cannot put on weighing balances. Therefore, our goal was to understand the stomatal response, but in terms of water potential regulation. Now, the next question is why we selected pines. So pines, they are ecologically as well as economically very important genus. They are native to the northern temperate regions and they are dominant species because they cover 30% of the northern hemisphere. And in this study, I am basically focusing on Pinus radiata, which is the most widely planted softwood conifer species, and it is native to Californian coast in North America. Also, it is known for its fast growth and for its value in role of forest production. Now, we have a sad reality here, and that sad reality is drought stress. So drought stress is not only impacting the natural distribution areas of Pinus radiata, but it is also impacting the plantation areas in New Zealand and Australia. Let's talk about drought stress. So what causes this water stress in plants? So there are 2 main key drivers which actually cause water stress in plants. Number 1 is VPD, which is vapor pressure deficit, and number 2 is soil water availability. So when there is high atmospheric demand, the atmosphere tries to suck water out of the plant, which leads to higher transpiration. And this leads to drop in water potential, and as a result, the stomatal conductance decreases and leads to stomatal closure. When the water availability in soil is low, it also leads to drop in water potential and the stomatal conductance decreases, which leads to stomatal closure. So what we see is that stomatas, they actually close, and this way they try to reduce the water loss. And this helps in restricting the decline in water potential, which otherwise can lead to xylem damage. But all of this is coming at the cost of reduced carbon gain. Now, the question is, do all plants have similar water potential regulation strategies? So, different plant species or different plants can have different water potential regulation strategies. Some are categorized as isohydric, which means that their daytime water potential, it remains almost constant. And some of them are categorized as anisohydric, which have a weaker regulation of daytime water potential. Now, these water potential regulation strategies can be described in terms of by explaining the differences in the stomatal behavior. So it is very important to characterize the stomatal behavior, but there is difficulty in characterization of stomatal response. So that difficulty arises when different stomatal responses are observed between different species or even within a species. So this study is from 1979, which shows that different species, so if you look at, I would use a pointer. So if you look at these numbers, so these numbers are actually different species, and you would see that how the stomatas, they have responded differently in each species. Not only between different species, but there are some studies which have shown that even within a species, you can see that there is acclimatization in stomatal response, which leads to a different stomatal response in terms of VPD, in response to VPD. Now, this acclimation to atmospheric condition, it leads to plasticity. So plasticity is it actually happens when there are variations in environmental conditions and water stress, which leads to changes in structural traits and also sometimes in xylem anatomy. And this is all linked to hydraulic adjustments. And this ultimately enhances the water use efficiency and prevents embolism. But this plasticity can be a pain for breeding programs because it is actually altering the stomatal response, which can further complicate the response of trees to the future climatic change. So our major concern was that can we use this stomatal behavior as a trait for tree breeding or not? So for this, to answer this question, we actually wanted to investigate the aspects of stomatal response which remain fixed and the ones which show plasticity under changing VPD. For this, the aim of our experiment was to determine the stomatal response to VPD in Pinus radiata, and this study is published in Plant Cell and Environment, and today I'm going to talk in detail about this study. So firstly, I would talk about the methodology, that how we conducted this study. So this study aimed to characterize plasticity of stomatal response, and for which we gave our plants 3 different treatments, out of which 2 were temperature treatments under 18 degrees and 30 degrees, and 1 was a water deficit treatment. So these treatments were actually given to the plant so that they can acclimatize to different conditions, and we selected 2 temperatures and 1 water deficit condition, and the plants, they were exposed to these conditions for a period of 3 months. Later, we took these plants to a controlled growth chamber where we exposed them to different levels of VPD to see how the stomatas, they respond under those levels. Now, in terms of measurements, so we basically monitored the needle width with the help of optical dendrometers. So these optical dendrometers were attached to the mature needles of Pinus radiata, and later the needle width that we got from these optical dendrometers was used to infer the stem water potential. Also, for transpiration measurements, we use these balances. Now, coming to the results, so in terms of morphological traits, if you look at this picture, the first 2 plants, they are actually from the 18-degree treatment group. Next 2 are from the water stress group, and last 2 are from the well-watered 30 degrees group. So yes, we found some plasticity in terms of plant height because the plants under 18 degree, they had smaller height as compared to the other 2 treatment groups. But in terms of total leaf area and leaf mass per area, we found no differences. But this wasn't our fundamental question. Our fundamental question was, is there any plasticity in terms of physiological response or not? And to answer this question, we actually exposed our plants to 4 different states of VPD. If you look at this first state, it starts from around 0.3 to around 2.1 kPa. And we actually wanted to see that how the stem water potential on this left y-axis and transpiration on this right y-axis, it co-varies in response to increasing VPD. So what we found was that with increase in VPD, the stem water potential here, it's actually declining in response to increasing transpiration rates. And if you look at these 3 colors, so these 3 colors are basically, they represent 1 plant from each treatment group. Also, we found that the water stressed group, if you look at this red plant, so this is the plant from water stress group, it had the lowest transpiration rate as compared to the other 2 treatment groups. And also the water stress treatment group, it had a slightly more negative water potential at all the stages of VPD as compared to the other two treatment groups. To study about the water potential in detail, we actually wanted to see how the stem water potential gradient, soil to stem water potential gradient, it varies to different states of VPD. So this water potential gradient is actually the difference between the soil and the stem water potential. And if you look at this figure, we have stem water, the gradient on y-axis and increasing VPD on x-axis. So overall, we found that all three treatment groups, which are in three different colors, they have a very conserved response to different VPD states. But if you look at the overall response, you would see that firstly, the water potential gradient, it actually increased proportionately with increasing VPD until a certain point, and later you would see that this later the response is somewhat linear. So this is due to stomatal closure. Now, the next thing is, what is this threshold? So this threshold is actually the stomatal closure point, or what we call as VPD breakpoint. And if you look at this figure, the three dotted lines, they are representing the three stomatal closure points in three different treatment groups. And the magnitude of difference between these three stomatal closure points, it was very small, which was just around 0.2 kPa. Therefore, we pooled the data together for all three treatment groups, and we tried to create a single breakpoint function, which you can see in this figure. And later, we have used this function to model the stomatal response to VPD. Talking about the stomatal response, so in this figure, we basically have the measured and the modeled response. The measured response is from the balances, which you would see as dot points, and the modeled response are these solid lines, which have fitted very well on the measured response. And this modeled GS is predicted from the delta psi dynamics that we have discussed in the last slide. So if you look at this response, you would actually see that there is a two-phase stomatal response. So you would see that there is low sensitivity, low stomatal sensitivity before this breakpoint, and later the stomatal sensitivity increases. But overall, you would see that all three treatment groups in three different colors, they have the overall response is actually conserved. So the difference that we have found is in terms of maximum stomatal conductance. So if you look at the maximum stomatal conductance in this blue group, which is the 18-degree treatment group, it is far higher as compared to the other two treatment groups. Now, the question was that what led to changes in the maximum stomatal conductance? And that was the hydraulic conductance. So the hydraulic conductivity of three different treatment groups, it differed significantly, and we found that it was maximum in 18-degree treatment group and minimum in case of water stress group. So overall, we found that this stomatal conductance, maximum stomatal conductance, it was very, very much positively related to the hydraulic conductivity across all three treatment groups. So we can say that we have found some differences in terms of soil to stem hydraulic conductivity, which were due to the adaptation of the plants to different treatment groups. And these differences in hydraulic conductance, they actually led to changes in the maximum stomatal conductance. So overall, we found that there was significant plasticity in terms of maximum stomatal conductance. But if you look at the overall response of overall stomatal response, it was fixed, it was conserved, and also the water potential gradient response was highly conserved. So I would conclude this by saying that yes, we can actually characterize the stomatal response using the water potential gradient dynamics. And the dynamic response, stomatal response is fixed in Pinus radiata, but if you look at the maximum stomatal conductance, that was plastic. Therefore, we actually need to account for this plasticity so that we can actually precisely predict the stomatal plant responses to future climatic changes. Overall, my study is a part of a bigger project, which is using remote sensing and artificial intelligence to develop new ways of phenotyping forest trees. Thank you very much. So at last, I would like to acknowledge my supervisors, my funders, and my lab mates. And to you all, thank you very much for your attention.